Hi, everyone. Wonderful to see all of you. Good morning. Hi, Nicola. Okay, cool. So, as we're still waiting for a few more folks to trickle in, I just want to welcome you to Foresight's Biotech and Health Extension Group, sponsored by 100 Plus Capital. Really happy to see so many of you here. This is one of the meetings where we talk about new technical need that's coming up on the pipeline. We are also having the policy discussions and investor Q&As. So those will be the next meeting in this series, and then we'll cycle through month by month again. So really happy to be joined today by Michael Fossel. Before we'll head into the presentation, I just wanted to make those of you who may not have been here last time aware of our longevity tech tree tools and prizes biotech workshop that is joining. That will be happening on May 21st to 22nd at IndieBio headquarters in San Francisco. And this will be one of our technical meetings. Usually this group emerged from our technical competitions, in which we usually try to just combine a few of our best scientists in the group to really work on the more long-term challenges ahead. You can tell we already have a really stellar cast, and with many of you who are in this call joining. So really excited to see all of you in person again. But the big hope for this, at least for me, is that these seminars are really incredible. I think to have a continuous update on what kind of like the latest and greatest research is in the field, but then actually to make progress on the more long-term problems, that would be really nice to also meet in person again. So I'm very, very excited to welcome many of you. In case you're interested in joining, there's an application form. Just say you're in the group, and that will, I think that would do the trick for many. So this will be shared sometime this week as well, but I just want to make the group aware, and this is a nice way to get in person again. Okay, wonderful. So enough from me from this end. I'm really, really excited today to have Michael Fossel here with us from Telocyte, and he'll be discussing aging, understanding it, and reversing it. Michael will be speaking for about 20 minutes or so. Then we'll have a Q&A, and I think you know the drill. Dump your questions in the chat if you want to make sure that they get that they can get selected first, and otherwise just raise your hand at the end, and yeah, feel free to have an open discussion in the chat. We are really excited to have Michael on board, and we've been actually reaching out to you, I think, already last year. So very excited this is finally happening. I'm sure that you have a ton of really exciting new results to show. And with that being said, welcome to Foresight's Biotech and Health Extension Group, and the stage is yours.
Thank you very much. I'm going to literally take the stage. I'll try to move quickly. I usually speak far too quickly for most people, so put up with me, but let's see what I can get done. I'm going to talk about understanding aging as a prerequisite to actually reversing it, because that's literally what I'll be talking about. There are three things I'm going to go over. One is sort of the illusion of understanding. We often think we understand something and don't. Secondly, I want to focus on how to actually understand the aging process, and then reversing it at the most fundamental level, the cellular, the genetic, and epigenetic levels. There are a number of times we think we understand things. A classic was Avicenna, whose medical textbook was the height of the world for 600 years. He understood the plague in the sense of understanding how to identify it, but had no idea about microbial disease. Vatican astronomers 500 years ago, excellent knowledge of planetary positions, no understanding of orbital mechanics, and so house arrest for Galileo. Lord Kelvin, 8 years before the Wright brothers proved him wrong, emphatically said, we will never have heavier than air aircraft. But it wasn't that he was ignorant. It was just that he didn't understand things. And that's often true. And here's another example. Back in the 60s, when I first was taking geometry and calculus, I found most people knew the formula for example, the surface area of a sphere, but they didn't understand it. And I found that I could simply use integral calculus to rederive it on a test. And it was a little slower, but I understood the process. The same really is true of aging. Example, these are common clinical biomarkers of aging. They are not aging. They're outcomes of aging clinically, but by themselves, they're not aging. They're simply things that happen because of the aging process. The same is true if we look at cellular biomarkers, whether we're looking at telomere length, ATP changes, methylation changes, the Horvath clog. None of those are aging. They are clinical biomarkers, but they don't tell you how all of these things interrelate dynamically and what the process is. Even if we go down a level and look at genetics, I've been focusing strategically on age-related dementias for the last year or so for good reason. These are more than 200 genes I can list, there's just some of them, that are correlated with aging, and in this case, age-related dementias, but they are not dementias. whether I'm looking at ApoE4, ApoE2, or any of the presenilins, those do not define Alzheimer's disease or any other dementia. However, they do tell you something about some upstream risk factors. This is a favorite curmudgeon friend of mine. He first described cell aging 61 years ago. He's still alive and well, by the way. He turns 94 in May. He and I still communicate. This is a favorite quote that I got from Len, and he is right. Almost all of us would say the single best predictive risk factor for aging, for age-related disease, is aging. But then almost everybody, academically, research, pharma, ignore aging and focus in on downstream clinical outcomes and upstream clinical risk factors without focusing on the aging process. I first described this, first talk ever in history, actually, here at the NIH 26 years ago. The talk was called Reversing Human Aging. And the point I was making is, again, if we could understand the aging process at the cellular, genetic, and epigenetic levels, the potential was there to intervene effectively in age-related diseases. I followed this up with the first 2 articles ever in the medical literature on exactly this same issue. Can we actually intervene in age-related processes? That was followed by this book, first book ever out on that topic as well. Followed by this, my Oxford University Press came out in 2004, looking at it in some academic detail. And Elsevier has just contacted me, asked me to please write this book, which will be coming out in a year or so. We're going to be looking through the most recent improvements in gene therapy and other interventions at the fundamental levels in aging. This, roughly speaking, what's going on, this is taken from an article I put out about a year or 2 ago that got more than 1,000 reprint requests, a lot of invitations to chair global conferences. I just got back from chairing a gene therapy conference in Boston. But the point I'm making here is that we're looking at upstream risk factors and downstream clinical outcomes and biomarkers. But what we need to do is understand how you get from one to the other. Why, for example, does a history of exposure to dioxin increase your risk for Parkinson's? Why do these things happen? Head injuries, increased history of encephalitides. Why do these things cause downstream risk, downstream outcomes clinically? Clinically, then, let me give you an example. If I'm looking at patients with Alzheimer's or dementias and other dementias, what I find is that typically my Alzheimer's patients, many of them will have motor deficits, typical Parkinson's. If I'm looking at my Parkinson's patients, many of them will have a variety of cognitive deficits, whether it's executive function, memory loss, or other cognitive deficits, typical of Alzheimer's. What you find is that while these clinical syndromes and frontotemporal and vascular dementias are all too real, there is overlap. There are gray zones. You find that almost none of the patients are entirely typical. And the same thing is true if I expand it to the alphabetic litany of other diseases, and I could add another dozen of these too. What you find is that almost no patient is entirely typical. Some patients early, some late, some have more executive function, some have more motor. They vary. A lot of them at autopsy will have vascular dementias as well. So they're mixed syndromes. Over about the past decade, a lot of us then have begun to suspect that your clinical outcome, the syndrome you end up with, depends on your clinical starting point, genetic, epigenetic, and behavioral history. But that they all play out on a common stage of age-related disease at the fundamental level, that is, in this case, cell aging. And that prompted the Alzheimer's Association to actually have me lead this conference on animal models in dementias, culminating in this paper that, as I say, got a remarkable reception. We're going to find that the same is true if I look at age-related cardiovascular disease. For example, I've seen myocardial infarction patients at age 25, very atypical, but it happens. Some atypical patients for CHF will have one syndrome, some another. Same for stroke, same for peripheral capillary pruning, but also many of these patients overlap. So again, if I look at sort of the alphabetic litany of various age-related cardiovascular diseases, you find some of the patients with varicosities also have aneurysms. Some with aneurysms also have hypertension. Some with hypertension have VTAC. So things vary. And again, what you're seeing is that there's almost no typical myocardial infarction. Some have pain, some have dizziness, some have shortness of breath as a presenting symptom. We're all a little different. And again, the suspicion is what we're looking at is your clinical syndrome, your age-related cardiovascular disease, is highly dependent on where you start. Do you have a history of smoking, for example? What is your genetic history, your epigenetic history? This matters. But that they all play out on a single common stage, which is cell aging. So this is a paper in progress that will be out later this year. The same is true sort of pari passu. If I look at other age-related diseases, I could just as well include here, for example, pneumocytes and idiopathic pulmonary fibrosis. But whether I'm looking at osteoarthritis, osteoporosis, chronic renal failure, immune senescence, all of these derive from one type, or in many cases, many types of a cell aging in various cell types. But the kicker is that aging is not ubiquitous. Every single one of you, as you're sitting there right now, if I will look carefully at your somatic cells, I'd find age-related changes. And yet you derive from an otherwise perfectly normal, given your genetic history, perfectly normal fertilized ovum. The ovum comes from your mother, a couple of decades old, from her mother, and that provenance goes back about 4.5 billion years. But what you note is that you did not inherit an aged cell, but your cells now age. So the question is, why the inflection? The same is true if I look at mitochondrial aging. What you find is that every mitochondria in your body and your somatic cells now, if I look at it in detail, and I'll get into this a little bit, I'll find some subtle but pervasive changes. But you did not inherit aged mitochondria. And that provenance goes back, again, almost 2 billion years, depending on who's doing the estimates. In fact, I recently was chairing a conference on mitochondrial aging. And on the panel, these are kind of the major questions that some of the audience asked. And they're good questions. If mitochondrial aging is the be-all and end-all of aging, why didn't it occur retrospectively? Or the obverse question, which is, why do mitochondria progressively fail and they do only post-fertilization? Example. These are a couple examples of this. These are taken from some classic papers from a friend of mine, Ed Byrne. Many of you may know him. He's the classic big name in mitochondrial cell aging. But these are just papers of him from 20 years ago. He's written 100 since. There are probably about 100,000 papers plus on this by now. And these are only some of the examples. But what you also find is that aging occurs not because of mitochondrial function, but something more fundamental. So I want to go back and look at aging in a much more fundamental way. So let me introduce you to some of my favorite species. Human beings, typically, mean lifespan now runs around 3/4 of a century. That's Coco in the upper left. I am probably the only person you'll ever meet who has been bitten by, wrestled with, and kissed a lowland gorilla. That's Coco. I was her babysitter for a year at Stanford years ago. She died at about age 40, and I will miss her. Typical domestic animals, dogs and cats, lifespans 15, 20 years, again, highly dependent on breed and species. Typical rodents, you're looking at 2 or 3 years, again, dependent on breed and species. So the immediate thing you'll notice is you cannot simply ascribe aging to entropy without explaining why entropy works at different rates in different species. Now, most of us at this point would sort of wave our hands and say it's genetic, which is true in a sense. For example, you share about 75% of protein encoding genes with that mouse. So most of us would say the reason you age at 40 times slower rate is because of the other 25%, to say nothing of the non-coding elements involved. But that's not an explanation. All you've done is wave your hands and said it's genetic. The same thing, by the way, pertains when you're looking at a single species and a single gene. These are Hutchinson-Gilford progenitor kids. 20 years ago, I was gathering together annually from around the world. Typically, we'd have 30, as you have here. In this case, I've just given them a tour of the White House, going places you can't go. All of the kids in this picture are dead now. They die at average age of 12.7 years, overwhelmingly of age-related diseases, typically strokes and heart attacks. The question is why? We know the gene, that is, it's a lamin A gene and progerin, but that doesn't tell you how it works. Why do these kids age 5 or 6 times faster than you and I do? A couple of my articles down there, and a really nice recent article showing we can actually reverse this. This is an article from December showing we can actually reverse the aging in their vascular endothelial cells. A friend of mine, John Cook at Houston Methodist. Overall, what's going on is not just entropy, but a failure of maintenance. If you look at any molecular pool in the body, you name it, any of them, with proteins, lipids, glycolipids, you find that the turnover rate in young cells is faster, so they tend to wash out damage. This is not as true of older cells, so they tend to accrue more. That formula is taken from my textbook, and you can find it there if you'd like it. But let me give you an example of this. If I'm looking at the central nervous system and focusing solely on beta amyloid for a minute and look at microglia, what you find is they produce it, they secrete it, they rebind it, they internalize it, they degrade it, and this is a continual recycling process. All of those processes have been measured, and all of them show slowing as microglial aging progresses. So you begin to get microaggregates. If you are really lucky and you've got a good genetic background, for example, you're biallelic for ApoE2, what you find is you don't make very sticky beta amyloid molecules, so your rate of accrual is low, even though as you get older. So some of these patients, for example, with a biallelic ApoE2 will not have Alzheimer's or microaggregates even at age 90. However, if you're unlucky at the other extreme and are biallelic for ApoE4, you make sticky molecules. And so these patients begin to show beta amyloid microaggregates even in their 20s and 30s. And in all cases, as we begin to get older, you begin to get these beta amyloid plaques. This is not the be-all and end-all of Alzheimer's, let alone other dementias, let alone other age-related diseases. But if I extend that within Alzheimer's to tau proteins, neurofilament light chains, I see the same thing. There's a slower rate of turnover and accrual of damage. The same is true of mitochondria generically, as well as DNA repair. Example, if I'm looking at this case, this is a cytochrome C oxidase molecule model. And what you find is that's being turned over more slowly. 99% of them, the aerobic enzymes in your mitochondria derive from the nucleus, and we see these as telomeres shorten. We see a slower rate of turnover of these. And that's true of almost all the aerobic enzymes we look at in the mitochondria. So not only do you get this decrease of ATP and increase in free radicals, but the membranes leak as well. So you begin to find a highly porous membrane, not only in the mitochondria, but the nuclear membrane. So you're getting damage to the DNA, but also they're generally scavenged, the free radicals. But the scavengers, in this case, you're looking at catalase, but it could have been one of the superoxide dismutases or any number of others, are also being turned over more slowly. So you make more free radicals, you leak them into the cytoplasm, you don't scavenge them as rapidly, and when there's damage, you don't repair that as well. Almost every molecule in your body you turn over, with one kind of exception, and that is DNA. But if we look at the 4 major families of DNA repair enzymes, what you find is all 4 of them also show slowing in cell aging. All of this is set by telomere shortening and changes in epigenetic pattern. These are exponential increases because each of these is independent. So you see an exponential increase in cancer over a lifespan, whether it's a 2-year-old mouse or an 80-year-old human. It is not exposure to carcinogens as much as it is the rate at which you're able to repair the damage from the exposure to carcinogens. And that goes down with cell aging. So overall, what you're looking at is cell division, telomere shortening, absolutely necessary changes in gene expression, which we have measured, slower telomere, slower molecular turnover. Therefore, you get increased molecular dysfunction, increased cellular dysfunction, and what I see clinically as age-related diseases. A couple of points I want to make. One is that telomere length is completely irrelevant. Absolute telomere length is irrelevant, but relative telomere length is a critical issue because it changes the pattern of gene expression. That's why there are mice out there that have telomeres 10 times longer than mine at birth and lifespans 40 times shorter. And yet, you can accurately predict the lifespan of almost any species based on the rate at which they lose telomere segments. So the issue is not telomere length. Ultimately, it's the pattern of gene expression, which is set by the rate at which you lose telomeres. This has been looked at fairly in fair detail. Some of the articles are quoted right down there below. Second thing I want to point out is that almost every article I've seen in the past 10 years on telomere measures is silly. I was going to say stupid, but I'm trying to be polite. It's silly. It's naive because you're looking at leukocyte telomeres, which have nothing to do with what you want to measure. I first pointed this out in the literature 10 years ago. Here's another nice article from just this last month saying the same thing. If I want to find out whether you've got osteoarthritis, I don't measure peripheral telomeres. I measure chondrocyte telomeres. If I want to find out, it would be like determining whether I've got osteoporosis on the basis of whether I have gray hair or not. Temporal correlation, but a hell of a poor way to make a diagnosis. So, but it's understandable. It's so much easier to get your blood samples than it is for me to get a brain biopsy for you, let alone coronary artery samples, let alone other things. But it messes up the literature badly. So almost all the literature out there that looks at telomere lengths and aging is looking at something that has nothing to do with disease. Next thing I want to point out is almost all the literature talks about replicative senescence, not really cell aging. Cell aging is a lifelong process. You see these shifts in epigenetic pattern throughout your life as cells begin to divide. Replicative senescence is a terminal event, and it's a mistake to confuse the two. They are related, but they're not the same issue, particularly if you're discussing things like senolytics. Now I want to talk about the possibility of reversing it. If we look upstream and try to affect cell aging, what you find is that none of the things we intervene with in the upstream risk factors ultimately can stop, let alone reverse aging or age-related diseases. So yes, it is useful for me to make sure my patients are euglycemic and normotensive, for example, lowering their incidence of age-related cardiovascular disease, but it doesn't stop it, let alone reverse it. The same is true of downstream risk factors. The classic is the monoclonal antibody study like solanezumab and anakinumab and so on. What you find is that you get this temporal shift, but then there's a parallel vector on disease going downhill to the placebo. So you're not actually showing you can stop, let alone reverse, age-related neurodegenerative diseases. However, we can do this with cell aging. Here's an example. This study came out in Science now 24 years ago. Andrea Bodnar, her group out at Jaron in California, showed that you can use telomerase to reset human fibroblasts in vitro and show that not only do you get the same normal pattern of young epigenetic expression, but you get normal morphology, histology, and physiology. The same thing is true, it turns out, when you look at age-related changes in this case, in tissue. This was a great paper by Walter Funk, hard to read paper. I actually had to take Walter out for a drink, then go back and look at his slides to get it right. But what he shows is you can actually grow young human skin from old human skin cells as long as you reset telomere lengths and reset the pattern of epigenetic expression. The same thing turns out to be true of osteocytes. The same thing turns out to be true of vascular endothelial cells. And this has just been redone, as I said, by John Cook last month at Houston Methodist. He used mRNA. In this case, that we're actually using telomeres directly. The same thing is true of organisms. This is not the first study that looked at this, but this is the best one from the clinical perspective. This is Maria Blasco's work in Madrid. She used an AV9 vector, a CMV promoter, and a mouse TERT. And not only do you get a lifespan increase, but you get normalization of behavior as well as improvements in bone mineral density, subcutaneous fat, immune response, and other things. The question is, how effectively can we do this, which is a different issue. So overall, what I want to do is point out to you that one, the upstream risk factors are consistent with a model looking at cell aging. Two, the downstream biomarkers likewise are consistent with our understanding of cell aging. We can understand why all those downstream biomarkers clinically and otherwise occur, as well as the upstream risk factors, why they cause this. And also, this model explains the heterogeneity both within and between species. So, for example, we begin to understand why I get Alzheimer's, you get Parkinson's, why she gets Parkinson's early and the other person gets it late. We begin to understand this, as well as the upstream risk factors, as I say, exposure to dioxin or paraquat, history of encephalitis. But it's also true between species. We begin to understand why dogs do it slightly differently than mice. Dogs are a little more similar to humans in terms of beta amyloid, but we begin to understand why species have this heterogeneity based on this model. The model is also consistent with all the research and clinical data as long as you're a little careful to understand what telomeres are doing and what they're not doing and how to measure them and how not to measure them. It also explains the mitochondrial changes because we can reset them. It's also been shown to be predictively valid. For example, the Eli Lilly studies, the Biogen Eisai studies, the data was predicted ahead of time based on the same model, and we get it right. More importantly, maybe it offers us a novel practical target. It's been shown to be effective in vitro, in vivo, and ex vivo, and it's a feasible technique. We can now take the same sort of technique that we used through SMA, for example, in the classic Avexis work, and use the same thing to go after Alzheimer's. In fact, the person who designed the Avexis work was initially on our board. So let me ask two final questions. The first is, can we reverse cell and tissue aging? And there the answer is yes. That had already been done. It's been done for the past 2 decades, and there are many more studies looking at this since. The question is, have you looked at it carefully in terms of what telomeres really do, what they don't do, and what gene expression is changing and how? The second is a more critical question, which is, can we reverse cell and tissue aging in human clinical trials? And there the answer is, we almost certainly can. The data suggest we can. We soon will. I just chaired a conference on gene therapy. There are a lot of fascinating new abilities to do this, not only with viral vectors, but some other interesting approaches that we're going to take to human trials sometime in the next 2 years. So I'd like to thank you all and remind you, please, that what we should be going after in life is not the little stuff. It's not simply a matter of treating biomarkers. As Lee Bai said, we should be hunting the tigers and dragons, not the rabbits. Thanks. I did that in 12 and a half minutes. Geez, Allison.
Oh, very fast. Wonderful. Okay, you already have
let's do
lots
that.
of questions.
1 minute.
You have lots of questions stacked up here already. Cool. Okay, thanks so much. Okay, quite a lot to digest here, and people are already digesting. So first one up, we have Marina with the question.
Thank you, Alison, and thank you, Michael, for the great talk. I was really reading this information about the telomere lengths in the leukocytes and kind of knew this information, but I was always unsure. Thanks a lot for clarification again. My question is, I'm doing the dementia research in humans. And my question is, do you have any guess on how can we measure telomeres in brain, in neurons? And the second part is there any relation between the telomere lengths in neurons and astrocytes, any other glial cells?
Yes, to answer your second question first, there's a relationship. One, there's not much data as you might like to have for, I think, obvious reasons, which most people don't like to give brain biopsies. And that will take us back to your first question. But it's hard to understand the correlation between neurons and microglia, for example, and astrocytes, as well as other glial and subtypes of glial cells. And you might initially say, well, what do you expect with neurons since they don't divide? And the answer is, well, they sort of do. And incidentally, in the classic study from Ron DePinto at Harvard, you find that you can actually reset both neuronal stem cell function and proliferation and differentiation, neural stem cells. So we know that there are neural stem cells. To some extent, they're important, and that we can reset them. But you're right. In the easiest way to answer your first question is to say, no, you can't. You can do a brain biopsy, but there is seldom justification for that ethically or medically. However, there are other things we can do and are looking at, and they are, maybe I should say, not ready for prime time, but they involve looking at a number of enzymes and also genes, DNA samples, and so on, in, for example, CSF. And that's a little easier for us to get. Most of my patients are a little more likely to let us do an LP than to do a brain biopsy. But still, the quick answer is, if I've got a patient who's got Alzheimer's and I'm treating them over a 6-month period and following it, I can follow blood, I can probably follow CSF, but I can't actually follow what I'd like to do as one of the primary biomarkers, surrogate biomarkers.
Thank you.
Okay, wonderful. And then next one up, we have, I think, Aaron with a few questions.
Yeah, thanks for coming. So first one is, it's difficult to get reliable measurements of telomeres. And I was wondering if there's any progress on that. Like, there's no baseline for telomere fluctuation over time. Do we have that yet, or is that?
You bring up another issue, which I didn't even bother bringing up because, again, I was trying to fit this into 20 minutes. But the first problem is that people measure peripheral leukocytes when they shouldn't be. And I'll come back to why even that's not a good idea. Secondly, when they measure them, they tend to look at things like mean telomere length rather than shortest decile, which is a much better predictor, or even subsections of that decile. We should be looking at short telomere lengths, not mean telomere lengths, because they tend to decide how that pattern is going to look in terms of epigenetic expression. But there's a much more interesting issue, which is that if I look at your peripheral leukocytes, they're very responsive to a number of inputs. So, for example, I'll give you a silly example. If I look at your mean telomere lengths right now in your leukocytes, and let's say that it's 10 kilobase pairs for the sake of argument, and right now what I do is I give you a severe viral infection, maybe COVID. I add Ebola if you'd like to. You lose your job, your dog gets killed, your spouse leaves you, whoever they are, your house burns down. You'll be under an enormous amount of stress. And if I measure your telomere lengths, say it is now 9 kilobase pairs. Not surprising. But if I look at the marrow, the hematopoietic stem cells behind those, what I find is that the shift has been almost not at all. Because what's happening is the cells are dividing a little bit in the marrow, depending on whether you're looking at T and B cells, and a little bit peripherally T or B cells. And so they're responsive to the stress. So it's also true that if I cure your COVID, cure your other infections, you get another job, your spouse comes back, your house is rebuilt, you make $1 million, suddenly you'll find your telomere lengths peripherally are a lot higher. But it is not because of reversed aging. It's because they're not churning peripherally. If I'm looking at the more important marrow component, you find that hasn't happened. I'll give you an analogy. This would be like, let's say I go to New York City and I go to, for example, Brooklyn. And I go to Brooklyn 50 years ago where there was a bad area. And I ask myself, what is the average lifespan, the average age of a person living in this particular section of Brooklyn? And I find that it is actually 75. I've got a lot of retired World War II vets or something. And now I go back and I measure it. And now a lot of people have moved in. The drug problem is cleaned up. You've got young people with kids. And now the average lifespan the average age of those people in that same city block is now 27. Have I made them younger? No, not a bit. All I've done is I've seen a demographic shift. And this is the same thing that's going on when I measure peripheral leukocytes. So, what you find is these articles that will say, listen, I have patients, here's their peripheral leukocyte measures. I put them on a vegan diet. I taught them to meditate. I did a lot of other things, a lot of other changes to the lifestyle. And lo and behold, the telomeres are longer. It means nothing. It's, again, sort of like trying to assume that because we've done something in the way of urban renewal in one city block in Brooklyn, therefore the average lifespan of human beings throughout the planet has gone up. No, it's just that one city block. You're just looking at local churning. So most of the articles on this are misleading. Let me just put it differently. The data is good. The interpretation is wrong.
Right. So do we have any longitudinal data from like a marrow or some kind of stable set of tissue that says, okay, here's how the telomeres fluctuate over time. Here's the baseline. And then because right now, if you try to do any kind of therapy, you don't know whether it's due to natural fluctuations or from the therapy itself. So is there a baseline data set out there?
Well, if you're looking at a well-defined, for example, a well-defined stem cell population, it's a slightly different matter. So, for example, your hematopoietic stem cells do express telomerase transiently, and they partially reset telomere lengths, which is why we're not all anemic at age 100. We've got other problems. So you do find that, but there's a steady, slow drift downhill. And that's not true when you're looking at N cell populations like peripheral leukocytes. So if you look carefully, for example, in animal studies, and they're looking at hematopoietic stem cells, you can get some baseline data that looks pretty good. You can see that telomeres don't so much fluctuate, more or less, but they show this slow decline. So it depends where you're looking and how you're looking. If you did this with human marrow, I expect you'd see the same thing, but it's a little hard to get human samples sort of every 6 months for the rest of your life. People, again, I don't know if you've ever had a marrow biopsy, but they're not comfortable.
Okay. All right. So then I guess talked about telomerase a little bit, but so it exists in human stem cells already. And I guess the question is, well, yes and no. Sorry.
Well, putting it simplistically, yes. I mean, actually, what you find, usually there's a constituent expression of H-TER, but not H-TERT, and there's this transient re-expression of H-TERT. But forgetting all about that, the answer is, yeah, kind of.
Okay.
Right. I don't mean to get too technical, so.
Okay. All right. That's it for me then.
Wonderful. Next one up, we have Nicola.
No, you're on mute still, Nicola. I can see you talking, but I can't hear you.
Sorry, I clicked and might
I was
have
at this
happened.
conference in Boston I was chairing yesterday, and I had to remind people that they don't have mute buttons anymore when you're there in person.
I was just thinking about the great talk. It was really nice. I want to ask something which is a little bit relevant to Marina's question. So the telomerase is important for cells that do enter S phase, otherwise nothing happens. If a neuron is supposed to have a life duration which is very comparable to the lifespan of the individual, does it really matter at all what the telomere length in that neuron is if it's not supposed to renew anyways? What could renew is a niche of potentially stem cells in the brain that will need to cycle.
Right. And, if what we're looking at, for example, is a large pyramidal cell that goes from my right motor cortex down midway down my spinal cord, clearly that's not dividing, because the important thing in that pyramidal cell is the connections and its actual geographic location in a sense. So dividing it doesn't really help us reestablish the right connections. Where if you're dealing with small interneurons, that may be more likely, and we're dealing with microglia and other glial cells, excluding oligodendrocytes, the answer is, yeah, you see why it would be important. Let me preface this next remark by saying, you have any idea how much we don't know? Lot. Yeah, right. As somebody once said, I know more about human aging than anyone on the planet. If that were true, I still know 1,000th as much as people need to know to understand what's really going on, because we don't. Sometimes we think we do, and it's a mistake. But we know the microglia divide. We know the microglia show not only telomere shortening, but these patterns of change in terms of molecular turnover of things like beta amyloid and other things, as well as mitochondrial dysfunction. But in many respects, the neuron is dependent upon those cells. They just are dependent physiologically. I don't mean just physically, in the glia sense, so that we recently talked about 100 years ago with early histology, but physiologically dependent. So if the microglia begin to be dysfunctional, we know that that'll have implications for the neuron, whether the telomere of the neuron shortens or not. So we know some telomeres shorten in some neurons. Many telomeres almost certainly don't. But we also know that there are other things that can cause telomere shortening rather than just cell division, as well as the epigenetic changes. As I say, telomeres are irrelevant. The question isn't what do telomeres do? The question is what's happening with gene expression. And gene expression changes independent of telomeres. So the complexities just become enormous. I sometimes like to wade through this by saying, I don't actually care what causes aging. What I want to know is, where's the single most effective point of intervention clinically and financially, which is a different question. And it looks like that point of intervention is to reset telomeres, but it's not telomeres I care about. It's the resetting that's critical.
You mind a second question?
Please.
Slightly relevant to your comment. I just would like to push you to another comment. In your initial slide, when you were speaking about the understanding of aging, you had on the left side measures of physiological markers. On the right hand side, you had the aging markers, and in the center, there was the cellular aging. This led the impression that you're speaking about the cell, one cell aging. Any comments about the importance of cell communication, and especially the ability of a cell to take in signals when it's aging?
Funny you should say that, because one of the favorite talks I had at this conference I led in Boston, some people have noticed something interesting, which is you can do DNA samples. You can do blood samples and look at DNA. That is, there's DNA in your blood, there's DNA that you give off, there's DNA in your feces. And somebody about a decade or two began to wonder if it's there on purpose. Is it just an accident, or does it have some function? And it looks like DNA, just like many other things, can be used for signaling between cells. More importantly, what you find is you look carefully, you find that these DNAs, in some sense, have zip codes. That is, they're targeting specific cells. So if I'm a cell and I'm sending out a DNA, I may target it to you, Nicola, rather than target it to Margareta. And it goes to you preferentially. We also know that that can have some effects on your cell function. So you begin to see why the model I presented is probably accurate, but nowhere near complete, not comprehensive. There's obviously a lot more going on that we still don't know about, in addition to what I've just said. Again, the historical analogies are classic. If I go back 500 years ago, healers knew more than I will ever know about small molecular approaches to treating a smallpox, but they knew nothing about microbial disease. There are surprises. An example of this is in gene therapy, most of the targets going for FDA now are aimed at orphan diseases like, for example, SMA. But the other thing you notice is that almost without any exception, not quite, they're all targeting protein encoding genes, which are about 1.2% of the human genome compared to at least 40%, depending on who's making the estimate, of regulatory components. And I don't just mean genes, I mean sequences and other things. So again, things are a lot more complicated than we think they are. There are interactions between cells that have to do with aging, and we tend to think of these simplistically. Gene therapy is going after protein encoding genes. No, that's just a start. We haven't even gotten going yet.
Great. Thank you. We have a few more folks in the queue. Next one up, we have Larry. You raised your hand.
Yeah, hi, Michael. I was wondering, have you seen, a lot of patients on heart therapy, of course, typically have shorter telomeres and things like that throughout, quite a bit. I mean, have you seen what you can attribute clinical consequences to that, that it can correlate, with aging or other things? I mean, some patients have been on them for 20 or 30 years and, from appearances, they don't look significantly aged, but are there other, clinical correlates that you can tie to, to those patients who have, you're typically, significantly shorter telomeres or relatively shorter telomeres?
Well, one, people age at different rates. I mean, I'm 71. Some people don't think I am. But we also age at different rates in different tissues and cell populations. So, I think we've all seen this with people we know who have osteoarthritis, but they don't have osteoporosis. If osteoporosis, but they don't have heart disease. If heart disease, they don't have Alzheimer's. They have Alzheimer's, but they don't have a lot of wrinkles. Different systems age, and it depends on, obviously, your lifespan. Your life history. So, for example, back to behavioral history. If I'm out and exposed to UV radiation all the time, I tend to get rapid skin aging. So it is enormously complicated. I'm still not answering your question, I have a feeling. But if you restrict yourself just to the cardiovascular system, you do see the sort of changes that you're implying. So, for example, if I look at samples, in this case, we're doing autopsy samples. This is 20 years ago, actually. You're doing samples from, for example, aortic arch, and you've got patients who have an aortic aneurysm, a dissection. What you find is they have a lot shorter telomeres than people who don't. The obvious question is this correlation or causation? Because people always confuse the two. You know, I like to say that the best predictor I know for what you buy in the grocery store for whether you've got heart disease is whether you buy infant diapers, because infant diapers appear to be protective against heart disease. But that's only because only 25-year-olds buy infant diapers. It's the correlation's not causation. But it's an intriguing correlation. And again, as I say, if we look carefully and try to reset it, that is, go in and intervene, we find that we can end up with normal endothelial vascular endothelial cells if we reset telomere length. The question is, does that carry over to human trials? So if you've got short telomeres in your aortic arch and are at high risk for an aneurysm or dissection, and I were to use, for example, an AV9 and pick my promoter and go in with H-TERT as an IV and reset telomere lengths, do I reset your risk of cardiovascular disease? An intriguing question. I bet the answer is yes, but as I always say to the graduate students and residents, good theory is one thing, but data wins every time, so we'll see.
Any follow-up, Larry, or?
No, that's fine, Allison. I mean, it's just that, you know, it's been an issue out there for a long time, and I haven't, you know, solid data that it's, you know, that there's significant deleterious effects from, you know, the inhibition of telomerase by these, by some of the antiviral therapies that are out there.
Yeah, almost, you know, my guess is, I was going to say almost certainly, but it's just a guess, that a lot of the things we look at that we think of as causing increased tissue risk work through telomere mechanisms. An example here would be toxins, namely, for example, oncotherapies. Clearly, they are toxins. And, you know, I used to say that it's really easy to cure cancer. I can do it, for example, with cyanide. Works fine. There are some side effects.
Sure. Yeah.
Same is true when you think of oncotherapies in general. They are, by definition, cellular poisons. The question is, can we preferentially poison oncology cells, the cancer cells, over normal cells? So it is, again, complicated. We know that any number of toxins, including oncotherapies, can end up with shorter telomeres for a number of reasons, not only by causing cell damage, cell replacement, and therefore shorter telomeres, but directly as well.
Okay, wonderful. Next one up, we have Abdul Kader.
Yes, hi. Can you hear me?
Man,
Okay, very good. My first question is about, for example, how the ECM induces a young ECM inducing an old cell and moving it into a younger stage, and vice versa. You have the case where actually the young cells changing also the ECM. Or you have old ECM inducing a young cell into becoming older. So that's my first question. And I
would have to stop you and ask you to clarify. Back in medical school, I actually started keeping a small book of abbreviations. For example, there are almost 20 abbreviations that come out MS, like mitral stenosis, morphine sulfate, and so on in medicine. And I find the same is often true of abbreviations. So if you're going to say ECM, please spell it out so if you haven't got the wrong ECM, because I'll bet you there are at least 12 of those as well. Please, sorry about that.
Yeah. It's these experiments have been actually quite successful using natural ECM.
Again, what do you mean by ECM? I just have to ask.
Extracellular matrix. Extracellular matrix. That's what I. Yeah.
I asked the question then again with regard to extracellular matrix, but I know that the extracellular matrix does the same thing. If I look at the collagen and elastin, for example, in my skin, it turns over faster at age 7 than it does now at age 71. So we know the matrix also does the same thing. Another example would be the extracellular matrix with regard to beta amyloid turnover outside of microglia in the intraneuronal space. So now, what about the extracellular matrix are you asking?
I think I don't recall exactly which type of cells and extracellular matrix were used. Those were in vitro experiments, and I can retrieve the references easily and send it to you. However, I think the more important question is that question. How consistent is cellular aging across the species in terms of rate determinant of aging? I mean, you're talking about a unified theory of aging for all species. This is my impression. So how much justified is this thought? Can you answer me that?
I can, but again, I'd like to give the caveat that there is, no matter what we think we know, we don't know nearly enough. So I'll just hark back to that one article I put out there it was a friend of mine now at Harvard, and the Whittemore article, when he was with Maria at CNIO in Madrid, looked at aging of animals and lifespan of animals. And as I say, there is a clear correlation. It's that nice curve, clear correlation between the rate at which you lose telomere segments and species lifespan. So that's probably the best. That's been duplicated in a number of other articles as well in the past 2-3 years.
Okay. Yeah.
But-
So
and again, I want to make that same sort of in-your-face argument, which is telomere length is completely irrelevant, but the rate of loss.
so you don't subscribe to the fact that different species may actually have a different mechanism of aging.
Not in that sense. I mean, this is the classic. You know, when we did that conference on degenerative or age-related dementias for Alzheimer's Association, the main point of that conference was animal models, and the question was, why does everything work in mice, nothing works in humans? And the answer is, most things don't work in mice either. But what you find is that if you have a genetic hybrid where you, for example, taken a human beta amyloid gene and put the requisite genes in the mouse, which doesn't use the same physiology, you find then when you do the research on mouse dementia, it doesn't translate well to human trials. And the answer is because one, you're using the non-native genes in the mouse, but two, even when you move the human genes in, they're still relying on physiologic pathways that are not present in the mouse. Because as I say, there are about 25% of protein encoding genes we don't share. And some of those are pathways that define how you treat beta amyloid. For example, the epigenetic pathways that define how fast you may turn it over, as well as producing the same beta amyloid. So it gets remarkably complicated. And looking at animal studies, what we find is that the fundamental mechanisms of, for example, telomere loss and cell aging are basically shared throughout almost all eukaryotes, depending on how you define the same fundamental mechanisms. What's also interesting is you begin to understand why it occurs. So what you find is that it makes sense in the sense of defining species lifespan to have a mechanism that not only responds to environmental change, for example, geologic change like global temperature or pH or oxygen, as well as biological change like prey-predator relationships. What you want is a very short lifespan to respond rapidly when environmental stress is enormous, because otherwise you die, the species is wiped out. On the other hand, you don't want it so fast that you end up with a high mutation rate. And you not only want it, if we have no mutations, we wouldn't be humans. If we had all the mutations, we'd all be dead. You want just enough mutations that the species can adapt to changes in the environment. But you not only want that small amount of change, but you want it to be able to adapt to the rate at which the environment changes. So it turns out this mechanism is not only fundamental for eukaryotes, but it actually makes sense in an evolutionary sense when you get down into it.
Yeah. My final comment is on that is the following, is that telomere shortening could be a cause or a correlation depending on the species. So
Well, that's what we thought back in the 90s. I mean, when Cal Harley put those first articles out at JERA, up until about 1998, and again with that classic Andrea Bodnar paper, where the thought was, yeah, it's maybe just correlational. Like I say, buying infant diapers, low correlation with heart disease, must be good. So the question obviously then becomes, what happens if you make people buy infant diapers and you find it doesn't make any difference? But what we found in regard to telomeres is that every time we reset telomeres in human and animal cells, we reset cellular function as well, and the cells begin to act like younger cells. So that makes a lot of sense going back to the classical Hayflick data. It makes sense in explaining, for example, why it is that you and I inherited perfectly functional mitochondria within your genetic background, perfectly functional fertilized ova, again, within your genetic background, rather than 4.5 billion-year-old cells. Because when you think about it, the truth is every one of us out there is 4.5 billion years old, and you look pretty good for your age.
Yeah, my critique was to telomeres, and I don't want to extend too much. I'm taking too much time.
Okay, final comment. Then we have
Final comment is that if it relates to telomeres, then actually what would happen is we would have seen a lot of variation of individuals because a mutation in telomere length is quite easy to achieve by cell. And I didn't find a good answer to that critique. So if you can, if really it's related just to telomeres, you would have seen a lot of variations easily by mutation in telomere lengths, which you don't observe in nature.
Well, actually, we do. I mean, here the classic example might be Werner syndrome, one of the adult onset progerias, or Hutchins-Gilford progerias, where we see it correlate very nicely both with cell function and with age-related disease in these individuals. The same is true of the other progeric syndromes. So let me again say, for one thing, that I don't really give a damn about telomeres. The question is, where's the most effective point of intervention? And to date, the data shows that telomeres are a very effective point of intervention. As long as, again, you understand the complexities involved, for example, measuring telomeres inappropriately versus correctly and not paying attention to telomere length, but changes in length. So if you use a sophisticated approach, telomeres work very nicely. And also to point out that there is a lot of variation. If I look at telomeres in almost any one of your cells, you'll find some of the telomeres on some of the genes are long, some are short. And people tend to, and again, as I said before, make the mistake of looking at mean telomere length rather than the shortest telomere length, which is what sets the pattern of gene expression. So these sort of complexities plague the entire field, and the response is, as a result, people draw silly conclusions. Like, as I say, they measure peripheral telomere lengths and then wonder why it doesn't clearly correlate with Alzheimer's. The answer is because you're measuring the wrong cells. I may as well measure your telomeres and ask how old I am.
All
Yeah,
right.
thank you.
We're going to wrap it up here. We have Oliver still with a question. Oliver, I don't know if you can unmute.
Oh yeah, it's pretty noisy here.
Hi, Oliver.
How's it going, Michael? Yeah, I guess about yeah, I might have missed it, but what's the roadmap in terms of time with phase 01?
The roadmap is a couple of things. One is that we're right now looking at a $25 million term sheet for Series A to take this to both the dog study and on to the NIH for human trials, specifically targeting Alzheimer's because it's our best strategic bet because it's fatal and there are no other treatments, anakinumab notwithstanding. There have been more than 2,236 interventional trials, all of which by global consensus have failed, just what we want, because I think we can show that we can actually reverse some of the cognitive decline in human beings, even in a phase 1 trial going over 6 months. So that's our near-term target. Now, the other question is, what do we use to do this? And in gene therapy right now, there are a number of viral vectors I could use. There are a number of promoters I could use. There are interesting interactions that we still haven't understood fully between the promoter and the viral vector serotype. So it's sort of like where we were in about 1930 with regard to air flight. We were beginning to learn a lot about aerodynamics, but we still didn't have the first transcontinental or transoceanic commercial flights going yet. We're still not quite there yet. We're getting there. Moreover, it's not at all clear that we need to use typical gene therapy with viral vectors these days. Now, this field is changing sort of week by week. No, we don't know what we're doing yet in many respects. So what we're going to do, the old tradition, you don't want to have the perfect get in the way of the good. What we're going to do is take our best approach and run it through probably a dual trial where we're looking, for example, in dog studies at least 2 different approaches and take the one that's the safest and the most efficacious to the FDA for the phase 1 human trial. And our first target, as I say, will be Alzheimer's. We're going to expand that out to other age-related dementias, Parkinson's especially. But then we're going to move to cardiovascular because it's actually a bigger market. It's just that there are alternatives, and not all cardiovascular disease needs to be fatal compared to Alzheimer's.
Wonderful. That always brings me to my last question, which is what, if people in this group are inspired and excited by your work, what can people in this group do to help move it along?
Prove me wrong. Again, back to that remark, a good theory is critical, but data wins every time. If somebody can prove, for example, that we can cure malignant melanoma with prayer and blue crystals, I may not think it's a great idea, but if it works, it works. But the opposite is true too. I may think this is a great idea. And the question is, can we make it happen? My nightmare is that the model will be correct, but we'll screw up somehow in trying to test it. So keep me honest. Keep asking these embarrassing questions like, how do you know? I don't. We'll see.
Okay, wonderful. Then another question that we often like to ask is if there's one kind of undervalued challenge, or like a challenge that you would really like to solve others in this field to make your research move faster, what would that be? So that's kind of the shopping list that that people can tell us. So there's one thing that someone else can work on, which would rapidly speed up your research. What would that be?
Yeah, it's delivery. Example, if I look at AAV9, which is a delightful viral vector in many ways, there are problems. One is in mice, it tends to transduce about 5% of the neurons and 1% of the astrocytes and less than that in the microglia, which is terrible. And yet there's a remarkable effect. So we're nowhere near good enough in terms of transduction. But at the same time, there are toxicities involved. For example, hepatitis, for example, DRG, dorsal root ganglia complications. There are immunogenicity problems. So if I, for example, give you an AAV9 and then I want to come back in a year and give it to you again, now you're alert, you have an immune response to it. How do I get through those sort of issues? So these are the things that are plaguing the entire field of gene therapy right now, not just what we're trying to do. But again, it's not as simple as just using gene therapy in the traditional sense of going in with a viral vector, whether it's lentivirus or AAV, because there are these other approaches too. So from my perspective, what we need is a reliable, safe, and effective way of delivering the kind of changes we need to make in genes, because ultimately, age-related disease is an epigenetic disease that is curable.